Okay, so now that we've talked about how we go from DNA into a protein, we've talked about gene expression. Now we want to start talking about why a cell may start to regulate that gene expression. So basically, deciding when to turn these genes on or off and why they might choose to do that, as well as how that happens. And so basically, what I want you to realize is that your body and the body of any multicellular organism pretty much is made up of a ton of different cells. And all of the cells inside of your body, with a few exceptions, have the same genetic information, right? And so your neurons, for instance, have the same genetic information as a macrophage or as an epithelial cell of some kind. You have all of these different cells that do different things, and yet they all have the exact same instructions in them. So it's kind of like if you had a book and somehow that book led you to read chapters 1 through 5, but your friend who had the same book had an entire different story because they read chapters 6 through 10. That's kind of how the DNA works with a cell. So, like, some cells are going to focus on some parts of the book, and other cells are going to focus on other parts. And that's going to become clear as we kind of start to explore this. But some genes are basically always going to be on, right? But most of the time, we don't want something to be on all the time. And that's primarily because if a gene is on, we're spending a lot of energy going through that transcription and translation process. That requires a lot of work. And so if you are trying to conserve your energy, you only want to turn things on when they should be on. All right. So this slide is kind of showing us a summary, if you will, of our central dogma of biology. So we have a single gene up top, right? This blue section of a chromosome. Okay. And then if we transcribe that gene, we will make a primary mRNA transcript. And then that's not quite ready yet because it has all those red sections. So we're going to now process it. We're going to cut out some introns, etc. We're going to cut stuff out, and then we have our actual mRNA strand that we're going to use. That needs to now leave the nucleus and enter the rest of the cell, the cytosol of the cell, the cytoplasm, right? And now we're going to actually make the protein at a ribosome. And so if we interrupt any part of this process, any part of this chain of events, we're going to interrupt the expression of that gene. And so we're going to see different ways by which we might do that. So the regulation of DNA, or rather DNA expression, is going to have a lot of different options, right? Because like here we had a lot of different steps. And so if we mess with any step, we're going to mess with the final product. So there are going to be some areas of DNA that will allow us to regulate the genes, even if it's not a part of the gene. There are going to be entire genes that are called regulatory genes that like code for proteins or allow us to manipulate other genes. We'll get to that in a second. And then we also have small regulatory RNA molecules that can interact with the DNA in some ways to either prevent or promote transcription. So the first thing I want to talk about is regulatory sequences. So regulatory would of course mean that you are regulating something, and a sequence is just a stretch of DNA, right? And so these are basically areas of DNA that are going to allow us to control transcription. We can turn transcription on or we can turn transcription off. Okay. And so there are three major types of these sequences that you need to be familiar with. We have promoters, we have terminators, and we have enhancers. And all of their names are called that for a reason. So all of their none of their names are trying to trick you. Okay, so a promoter is going to promote transcription. A terminator is going to terminate or end transcription. And so we're going to see each of these in action. But just know that in general, you're going to see areas around a gene. Okay, so here is our reading frame. So this would be like where we're actually reading the gene. We're going to see areas around that gene that allow us to turn it on and off. Okay? And so promoters, again, we are promoting transcription, so we're going to allow transcription to happen, are generally going to be upstream of a gene, excuse me. So like if we have a DNA molecule, right? And we are going from 5 prime here to 3 prime here. Here's my gene. This is my section of DNA that codes for a protein. The promoter is going to be upstream. Okay. And it's going to be nearby. It's not too far off. It's not technically part of the gene. Okay. Notice I have this little stretch of DNA in between. Okay. But it's nearby. Okay, just upstream. So this area is going to be really important with initiating or starting transcription of this gene. And so most of these genes are going to have a promoter. Not many genes are just on all the time, right? So most of them need to be allowed to be on by a promoter. So our next step is terminators. So terminators are going to terminate a gene or signal the end of a gene. And this is basically going to tell transcription to stop. Okay, so here we have a promoter upstream and a terminator downstream of a gene. So this yellow is now my gene. Okay, I have a promoter upstream. That's going to basically allow RNA polymerase to get on here. Okay, because remember, RNA polymerase needs like a specific place to bind. That's going to be where it binds. Okay. And then RNA polymerase is going to go from the 5' end to the 3' end, and it's going to transcribe all of this information. And then that's going to stop at the terminator. Okay. Now, sometimes you're going to see a terminator that is after a couple of strands of DNA. Okay. So just remember that we're after, and this is all relative. We're looking at the 5' to 3'. So if I have 5' on the left and 3' on the right, just remember that the promoter is upstream and the terminator is downstream. Okay, so the promoter is where RNA polymerase is leaving from, and the terminator is where it is going. Okay. Finally, we have enhancers. So these are like little sections of DNA that proteins called transcription factors can bind to. And basically, a transcription factor will bind onto an enhancer. So here's an enhancer. And it's basically like usually represented as this big old blob. And we're going to do a simulation in class on the Chromebooks that will allow you to see what how these transcription factors really work. But basically, they allow the promoter to be more accessible. Okay. It makes it more likely that RNA polymerase will bind on and then start moving. Okay. So it basically just enhances the likelihood of transcription, hence why we call them enhancers. Again, none of these terms are trying to trick you. Okay. So we also have regulatory genes. So these are entire genes. So they code for their own proteins. And so they might code for transcription factors. So like those big old blobs that I was just talking about, those had to come from somewhere. And they come from genes because they're just a protein. How do I make proteins? Well, I have to look at the instructions. I have to look at my cookbook, right? And so that has its own gene. But there's also little pieces of RNA called microRNA. So we have dubbed it miRNA. Okay. You can call it myRNA. Excuse me. And basically, they are going to allow us to access DNA or not. You don't need to worry about the specifics as to how they do that. You don't need to worry about how they bind, whether it's hydrogen bonds, anything like that. I just want you to know that these things allow us to turn genes on or off. That's really it. I don't want you to overthink this. Okay. So when we start looking at organisms like bacteria and viruses, we see gene expression with multiple means of control. Okay, we have a positive control mechanism and a negative control mechanism. So you can either induce a gene. So induce would be to like initiate it. So you like turning it on. Or you could have a gene that has to be repressed or stopped. So maybe it's on all the time until you stop it. Okay, so there's multiple options here. We're going to go through a couple of those. And then there's also regulatory proteins. So there's things that we can create within the cell that will allow transcription or prevent transcription, very much like we were just talking about. So often you're going to see diagrams of a gene being expressed with, random blob-like structures. And that's just because these are big old proteins. They're very complicated 3D structures. And so I don't want you to focus too much on that because they're almost always going to have a label. But sometimes you will have to be able to label it yourself. So you need to be able to recognize what something in the system is doing to the rest of the system, right? So like if you see something that is moving along the DNA strand from the 5' to the 3' direction, you need to be able to tell me like, oh, that's probably RNA polymerase because it's going down the railroad tracks and it's making a transcript, right? So if you see something making RNA, it's polymerizing RNA, RNA polymerase, right? So you need to be able to recognize that, as well as recognize certain things like, oh, this is where the gene started. So this is my start codon. Here's my stop codon because that's the end of the gene, right? So basically, some genes are going to be turned on by an inducer, or some genes are going to be on all the time unless you turn them off, unless you repress them with a repressor. And so basically, there are going to be two different kinds of operons, we're going to start calling them, that you need to be familiar with. And so if you only remember 2 slides from this lecture, this is the first one. Okay. So this is an inducible operon. So if you induce something, that means you're causing it to happen. Okay. It wouldn't happen without you inducing it. So like for instance, well, it might happen, but it would be a lot slower. So like for instance, if you think about inducing labor in a pregnant woman, that means that you're trying to get labor to start so that they can actually have the baby. Okay. So if this is an inducible operon, that means that it's not working. It's not doing its thing until you induce it, until you allow it to happen. So here I have a gene, okay, and on the left I have the gene not being expressed. So this is kind of where it's turned off on the left here, okay. So I have a promoter region and I have an operator. So the operator is like where the action actually happens, right? And then here we have the gene itself, the rest of the gene itself. And sometimes it'll be multiple genes if it is a complicated process. And we're going to go through some examples of that for both inducible and repressible operons. But here I have kind of the default of an inducible on the left. So the default for an inducible operon is it's turned off. And that's because something's blocking it. Okay, so here I have an active repressor. Okay, so something is stopping RNA polymerase from binding and doing its job. Okay, it's like a roadblock. So if you picture these as railroad tracks, right, you get on the station at the promoter, and then you're trying to go from 5' to 3'. You're trying to get to that terminator region. Well, you can't because there's this giant roadblock here. Okay? It's not until we remove that roadblock that RNA polymerase is able to go through and go all the way to the terminator region and create the mRNA transcript. Okay? And so the way by which we actually remove that roadblock is with what we call an inducer. So it's an inducing molecule. Okay? And so here it's just represented by this green ball, but it could be lots of different shapes, and you just need to be able to recognize what it's doing rather than memorizing a specific shape, right? Just like anytime we use these simplified diagrams of these real situations. So it'll basically bind onto that roadblock. Okay, so this red roadblock, it's bound to it, and then that causes it to leave. Okay, and now my roadblock is gone, and RNA polymerase can move. Okay, so just to clarify, just to summarize, an inducible operon, so it is inducible, meaning it is able to be induced, so it is able to be turned on, is normally off. Okay, the default is to have something in the way, it's to have a roadblock. So this will typically be a catabolic reaction. So like when you're breaking down something, because you don't want to try and break stuff down if that stuff isn't actually there. So we're going to go through an example of what that might look like in a real cell. So your repressor is normally active, and it's only once your inducer or your inducing molecule binds to and inactivates the repressor, so removes the roadblock, that the gene is actually able to be expressed. When that happens, the operon is turned on, is how we phrase that. Okay, so the gene or genes have been turned on. Okay. So an example of this is the classic example that you're going to see every single time pretty much is the lac operon. It's called that because of the sugar in lactose. Okay. So here's an example of what the lac operon looks like. Okay. It looks really, really complicated. It's not that bad. So if lactose is absent, so that means we don't have lactose, right? That means that our repressor, that's our red little Lego looking thing here, is active, meaning it's the roadblock is in the way. Okay. And thus my operon is off. I can't make my proteins. Okay. I don't want to try and make those proteins if I don't need them. Like if I have a protein that's going to let me break down the sugar from lactose, I don't want to make that protein unless there's lactose involved, because otherwise I'm just making it for no reason. So when you actually ingest some lactose, lactose is going to come along and it's going to bind to our repressor. Okay. And that's going to cause it to become inactive. It's going to cause it to leave. And then that allows my RNA polymerase to go downstream and create, in this case, a bunch of different proteins. So this is an operon that is made up of multiple genes. Okay, we have the LAC-Z, LAC-Y, and LAC-A genes. Okay, you don't need to memorize those or anything, but there's just multiple proteins that are going to be made as soon as this is allowed to happen, because genes are kind of like grouped together like that, right? Sometimes like LAC-Z is going to be useless unless we also do LAC-Y. So let's just do them all at once. Okay. And so we're going to make this big old mRNA strip that will have all three mRNA transcripts from those three genes. Okay. Now we're kind of flipping onto the other side of the coin. We're looking at the DNA strands that are normally on and need to be turned off. Okay, so this is like a negative control. Okay, so when there's no repressor present, it's just going to go and go and go and go. And that's what's happening on the bottom here. Okay, I'm going to transcribe and transcribe because nothing's in my way. There's no railroad blocks. Okay, but up here, I have a repressor. I have something in the way. So this is just like that red Lego looking molecule from before. Okay, and in fact, here's our red Lego friend. Okay, so this is slide number 2. Again, if you are only going to remember 2 slides from this lecture, this is the second one. Okay, so you have the inducible operon, and now we have the repressible one. So this operon is able to be repressed, hence the name. Okay, so in this case, it's happening all the time. It's always on. Okay, it just normally wants to be on, and so this is our norm. We're expressing this gene. Okay, but if a co-repressor, so something that represses together with the other repressor molecule. So this thing's like always around. This red Lego thing is always around, just like it was in the last one. It's always there, but it's not doing anything until a co-repressor, so something that represses together. Remember, things are named these for a reason. You need to get used to that if you're not yet. Okay, it's only once the co-repressor comes into the system that it will interact down here, and then that will allow the whole structure to bind to the operator and shut it down. Okay, that's going to repress this operon, make it so that it can't turn off, or excuse me, turn on. So normally a repressible operon is able to be repressed. You can only be repressed if you're expressing yourself, right? So it's always on until you turn it off. Okay, and so this is typically going to be for any anabolic metabolic pathways. So anytime you're building stuff, because building stuff is good, but sometimes you need to be able to shut it down. And we'll talk about some situations in which you might want to do that. So it's usually going to be an organic molecule that will be your co-repressor, and then that'll bind to your repressor. So that'll bind to your red Lego looking thing, and then that'll allow the repressor to activate, thus shutting down the operon, turning the operon off. So this would be how we turn the gene off in this case. So the most common example that you're going to see is the trip operon, or TRP. Okay, and that's for tryptophan. So basically, we have an operon. It's made up of lots of genes again. We have E, D, C, B, A. Okay, and we're going to make proteins E, D, C, B, A when we express those. Okay, so they're just going to go and go and go and go. This is RNA polymerase, and it's just going to go and go and go and go and make our mRNA until you introduce tryptophan. So tryptophan is found in some foods, right? You've probably heard it in the context of turkey, right? It's the thing that people think makes you sleepy from turkey. And so we have tryptophan here. And once tryptophan enters the system, it's going to bind to my red Lego thing, my repressor. And now my repressor is active. And once my repressor becomes active, it's the right shape. Because, like, notice here, if I tried to shove this shape into the railroad tracks, like, it's kind of angled weird. It's not going to fit. But if tryptophan binds, now it's this nice straight Lego looking piece, and so now it can shove right in, and it can shut down this operon. Okay. So just to kind of recap, operons are these big old sections of DNA that have just a single promoter. So there's only one section at the beginning, one promoter that's upstream of this set of genes that allows this whole thing to happen. Okay. And this operon is either going to be inducible or repressible. It's either always on or always off. This is just a different diagram to show the lac operon. Okay. So we have three genes, lac Z, lac Y, lac A, and that's going to code for three proteins if we express it all. Okay. And if lactose isn't present, then it's not going to be happening because we don't want to waste the energy. So this would, of course, be our inducible operon, just to recap. Okay. And it's always important to remember that this is happening in the context of a giant DNA molecule. This is not a small scale process, right? We're looking at small parts of a larger whole. Okay. Now, just to recap again, we have the opposite side of the coin. The lac operon was our inducible operon, but we also have the operons that can be turned off because they're always on. Okay. And so this would be our repressible operons. And in eukaryotes, this process is really, really complicated. And so that's why we'll often show a prokaryotic example because in general, there's just fewer molecules to keep track of. Okay. But basically, there's going to be a chain of events where something receives a signal and then they're going to send their own signal, and it's basically this whole complicated process, and it's just how a multicellular organism interacts with itself. So I don't need you to memorize any of the ways by which it does that, but I do want you to realize that there are ways that your cells control gene expression, and it's using that language, those vocab terms that we talked about before, so like transcription factors. So those are going to bind onto certain areas of DNA, and that's going to allow us to access certain genes over others. So this is just one way by which we turn genes on or off. Okay. And they're going to physically, this is important, physically bend our DNA. So here we have our DNA molecule, and then because this weird shaped thing has bound to it, it's now causing the entire DNA molecule to bend. So it's almost like how when DNA wraps around histones, it gets all condensed. It's really the same idea when DNA wraps around something that changes the physical shape. Okay. And so sometimes that's going to be necessary because in order for the other parts, so the transcription factors, mediator proteins, the promoter section, as etc. In order for all of those things to be in the right place at the right time, sometimes the DNA needs to bend. So like here, I have here an enhancer region. My enhancer region, if this was a stretched out DNA molecule, if it wasn't bent around this protein, it wouldn't be anywhere near the promoter, but bending it has allowed the promoter to get near the enhancer, and thus we can actually start transcribing. I don't need you to memorize any of that. I just want you to be familiar with it. So gene regulation, so that's turning genes on and off, is really, really important in every living organism, but it allows us to differentiate a lot. Okay, so it allows us to make cells different from one another. So, for instance, we have lots of different cells in our body, and you have a liver cell, you have kidney cells, you have brain cells, etc. Again, they all have the same DNA, and yet they turned out so different, and they do entirely different jobs. And that's because we turn different genes on and off. So, like, your liver cells have liver genes turned on. That's how they became liver cells. But your blood cells have blood cell genes. They don't want to turn on the liver genes. That's useless to them. Okay. And so there are different ways that your body tells a cell to become what it's going to become. And we'll get into that a little bit if we have the time. But what's really interesting is that we've been focusing on the genetic aspects of this for so long that it's really easy to forget that there are environmental factors as well. Your cells will respond to the environment and will respond to what environmental factors enter them, right? And so we saw that in the context of the lac operon. We saw that if lactose enters the cell, then gene expression is changed. But what's really interesting is when you get twins, so identical twins that grow up in significantly different environments, you can find significant differences in their bodies, right? And so just because they're twins doesn't mean they're going to end up the exact same. They can have different heights, they can have different facial structures. There's so many differences that can happen as a result of the environment. And it's just easy to forget that. I don't want you to memorize that or anything. I just want you to think, like, imagine a situation in which while you were growing up, somebody with the exact same DNA as you had worse circumstances. Maybe they were malnourished. Of course, they're not going to grow as tall as you. And so the environment has almost as large of an impact on your traits as your genes. And so it's almost always a combination. There's the genetic component and there's the environmental component, and they interact together to determine the phenotype. Okay. It's very rare for phenotype to just entirely be determined by genotype. Even when we were looking at Mendel's pea plants and whatnot, if the right conditions weren't in the soil, you're not going to get a green plant, right? They need to have everything in its proper place. So it's not just genetics, there's also environment. So here are some extra resources that I'm going to recommend. There's a really interesting field of biology called epigenetics, and there's a really good SciShow episode on that. But epigenetics is basically like your experiences change your DNA, and that's something that we used to think was ridiculous, right? So when we talk about evolution, we're going to talk about Lamarck a little bit, and he had this idea that basically giraffes got long necks because like the giraffe started stretching their necks and they needed to get higher and higher to get more and more leaves. But of course, we know now that that's not the case. We know that there were some giraffes that had longer necks and some giraffes that had shorter necks, and the longer ones just happened to do better. So they were more likely to survive and reproduce. But your experiences actually do change how your DNA is structured. So there are experiments where individuals have taken a rat and they've mated that rat. So now the rat has babies and the babies turn out normal. And then they take that same mother rat and they condition that rat to be afraid of a certain smell. So what they do is they introduce it to a smell and then they shock it. They do an electric shock. And so over time, they condition this rat, even if there's no shock involved, to fear and avoid that smell. And so then they take that same mother rat. So they had a litter before, right? They had kids before, and those kids turned out normal. Then they take that same mother rat after we've conditioned it to fear that smell, and she has kids again. And those kids are automatically, like, as soon as they are able to, be measured in a lab, they are afraid of that new smell. So this is one example of how epigenetics is so interesting. Your experiences shape your DNA in some ways. In this case, the rat's experience shaped its DNA, and so now its kids are afraid of a certain smell. And that's mostly an aside. I don't want you to focus too much on that. It's mainly just an interesting little anecdote, but I just don't want you to forget about the environmental impact. And so that's really all we need to worry about for Unit 6. That's all we really need to worry about for gene expression, is how we turn genes on, how we turn genes off, why we might want to do that, and what might affect gene expression. So like environmental impacts as well. So After this, we can move on into evolution and then finally into ecology after that.